to 3' exonuclease activity and polymerase activity, right? So this is the 3' end, right? So it's going to extend this piece of DNA this direction. But to do that, it's got to chew up the RNA as it goes. So it's going to chew that up at the same time that it extends this piece of DNA. But it can't attach these two pieces together, right? So DNA polymerase 1 is going to remove the primers and replace them with DNA nucleotides to extend and remove the RNA. Right, so same thing here. We're going to extend this and remove the RNA. Right, so we've taken care of one problem here. We've removed the RNA. Now, what about the RNA here and here? What's going to happen? We can chew it up, right? Can I build anything to replace it? No. Why not? You have to have a 3' OH hanging off over here to build onto. There's nothing there, right? It's the end. So, what's the problem here? Your DNA is get shortened, right? If we do replication again, it's going to get shorter. If we do replication again, it's going to get shorter. Every time we replicate a linear piece of DNA, the ends will get shorter because you can't replace that piece here using DNA polymerase. What is this region called at the end? Those are called telomeres, right? Right, so the sequence at the end is a repetitive sequence of DNA we call a telomere. The reason why we have this is it's kind of a buffer to keep the DNA from getting too short, because if I have a gene over here that is kind of important, right? If my DNA gets too short, I start to lose genes, right? That can be lethal. So you have extra DNA at the ends of your linear chromosomes called telomeres. Now, some cells, but not all cells, express an enzyme called telomerase. Right? Telomerase can fix that problem to keep the telomeres long. But only some cells express this gene. Right? When you're very early in development, right? Like 1 cell, 2 cell, 4 cells, 6 cells, 6 cell, 2, 4, 8, right? All your cells are going to be expressing telomerase because you're doing a lot of replication, right? You're making lots and lots of cells, right? But as you get older, most of the cells in your body start to stop expressing this gene. So one of the ways that cells age is because they stop expressing telomerase, and the telomeres over time get shorter and shorter. But if you're a stem cell, right, like a cell that's supposed to give rise to a bunch of other cells all the time, you're doing a lot of cell division, so you have to keep expressing this gene, right? What other cell type might decide, I want to turn this on so that I can divide forever? Cancer cells, right? A cancer cell typically finds a way to turn this gene on when it's not supposed to be on, so that it can continually express telomerase and keep the telomeres nice and long. If the telomeres get too short, your cells basically stop dividing permanently. Right? They'll initiate a program to keep them from ever dividing again. Okay, so that's dealing with the problem with linear ends. What else did we need to show here to finish up replication? Ligase, right? Ligase has to go in and link all of these pieces together. Questions? Okay. So I think that's it for replication. And then we mentioned repair very briefly. All right, if DNA polymerase makes a mistake and you have base pairs that can't match up because it put the wrong base in, right, the shape of the helix is distorted. So as a consequence, you have enzymes that can recognize that because the shape is different. So they go in and they basically cut on either side so that we remove a whole section of DNA. Right, so once I've removed this section here, right, then DNA polymerase 1 can come in and fill that. So if this is the 3' end, it's going to take this and it's going to fill in new nucleotides here. So did they show this right? Oh, I got my answer wrong. So this is the 3' end. Ah. Right, so here's the 3' end. So once these are removed, then we just stick in a new base, new base, new base. And then who's going to fill this in? Ligase will seal the gap. It's a really generic overview of DNA replication, DNA repair, right? There's lots of different types of repair, right? But the idea is if there's, for example, a mismatch, you have enzymes that can find it. And if we cut it out, we just use the same machinery that we use to do normal DNA replication to finish the repair job. All right. Okay, so let's talk about gene expression. Now you guys already learned the essentials for gene expression. You got like the overview, right? Because in lab we sat down and we did DNA replication, transcription, and translation. You did it on the worksheet, but you also did it with the model. How many of you guys have looked at the worksheet on the central dogma and mutations that it was posted? All right. So I highly, highly, highly recommend that you work through that worksheet in preparation for, which is on Monday. I'm not the only one. All right. I keep thinking what it is. All right, so let's start out here. First of all, if we're talking about gene expression, what do I mean by that? What does it mean if a gene is expressed? Okay, it might be that a protein is being made, so that implies what's happening. Transcription followed by translation. Okay, so I said it might be a protein. And the reason why I said that is because we have to kind of think about our definition of a gene. Generally, we almost always think of a gene as a region of DNA that codes for a protein. But that's not necessarily the case. Some genes just provide the information to make a functional molecule that doesn't get translated into protein. Can you think of something that would be a gene that would lead to a product, an RNA product, that's not going to become a protein? I mentioned a couple of examples. An RNA molecule that's not going to be translated to protein.
Is it RD primase?
Primase is an enzyme, it's a protein. What's in the ribosome? What's a ribosome made out of? rRNA. What is rRNA? Ribosomal RNA. Can you think of another type of RNA? tRNA. Can you name another type of RNA? mRNA. Okay, which of these is used to provide the instructions for protein? This one. these two, right, when we make the RNA molecule is the functional molecule, right? I'm not translating this. So I still have a gene, like an rRNA gene to produce this, even though this isn't going to get translated into protein, right? So we got to broaden our definition of a gene, right? A gene is really going to be something, a sequence in the DNA that codes for a protein or an RNA molecule that has some function in the cell. So make sure that you can make that distinction between the fact that it doesn't have to just be protein, it just has to be a functional molecule, so it could be protein or it could be something like an RNA molecule. Right, so what is that information? If I looked at a gene, the information is encoded in the sequence, right? Just like we would say that the information on a page is in the sequence of the letters, right? When you're reading, right, the reason why there's meaning to what you're reading is because of the order of the letters. In the same way, a gene, right, that information is in the sequence of the nucleotides. Right, now, we're going to focus on humans because that's the most relevant for us and usually pretty easy to remember. Our genome contains 3 billion base pairs. Right, that's how big our genome is. We've got roughly 20,000 protein coding genes. This doesn't include all the other genes that don't code for proteins. Right, now, here's the thing. Those 20,000 genes make up less than 2% of your DNA. Right, so I always draw like a chromosome like this, right? And then we're like, oh, here's a gene. That is not to scale, like even remotely close, right? Less than 2% of your DNA codes for protein. Right, so you've got a lot of DNA there, but very little of it actually codes for protein. So, when we talk about genes, those 20,000 genes, right, those are all protein coding. There's a few thousand more that code for RNA molecules, right, but most genes do code for proteins. Most, but not all. Right, so right now, what we're going to focus on is this transcription and translation process, right, but you understand that if we're making an RNA molecule that's just going to function, then we just stop at transcription. All right, so just to kind of put things in perspective, right? This is a generic sort of schematic diagram of a chromosome, right? This one is a duplicated chromosome. We'll talk about duplicating chromosomes later on, right? But this is chromosome 22, right? And so if we looked at, say, just this little region of chromosome 22, and we blew that up, right? This is 10% of that chromosome, and there's roughly 40 genes scattered along here. So these big red blocks represent genes. Now, the length of these things, remember we said there's 3 billion base pairs in the whole genome? How many chromosomes do we have in total? 46, 23 pairs. Right, so this is just one of the 23 chromosomes. But remember, they come in pairs, so I have 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, etc. Right, so the average length of these things is somewhere around 100 million nucleotides in length. Right, so and then you've got 46 of these things. If you add them all up, you get roughly 3 billion base pairs. Okay. So if we blew up this region, right, so we zoom in 10 times here, we zoom in 10 times again here, right, this is 1% of your chromosome, there's like 4 genes scattered across that. Right, and then when we look at one gene, there are multiple components to a gene, right? We've kept things very simple so far, right? But a gene is going to have some DNA sequences that are regulatory. For example, a promoter. This region is not transcribed or translated, but it's essential to the gene. Right? So it's not actually part of the gene, it's the regulatory part. So we'll talk about those on Thursday. And then in the gene itself, we have different regions called exons and introns if we're talking about eukaryotes, and we'll come back to this, right? So this is just to give you some idea of scale here. Eduardo?
What's the remaining 98% composed of?
It's all DNA,
But
it's
like,
just not genes. So you used to call it junk DNA, but that's really misleading, right? First of all, here's an example. Is this coding? No. It's important, but it's not coding, right? So there's lots of DNA, for example, that has some role, but it doesn't code for the protein, or it might not even code for anything, but it still plays some important role. So there's a lot of DNA where we don't know what it does yet, right? But it might do something like this. There's also a lot of repetitive DNA. So there's lots of different types of viruses. They're called retroviruses, or there's a few other different types. They literally copy themselves and insert themselves throughout the genome, right? So we have a lot of these scattered throughout. In fact, a significant amount of our DNA is made up from these viruses that have essentially made copies of themselves and inserted themselves into the DNA, done that multiple times. And then there's a lot of stuff where we don't know what it is yet. We just haven't characterized it. Okay, so on the left-hand side we have a prokaryote, and on the right-hand side we have a eukaryote. What's the obvious difference between these two when it comes to the process of gene expression?
The nucleus.
Yeah, there's no nucleus over here, right? Okay, so if you're a bacterial cell, or any other prokaryote, as soon as you make an RNA molecule, you can start the process of translation immediately. Because where are ribosomes found in the cell? Floating around in the
Cytoplasm.
cytoplasm, right? There's no barrier in between your mRNA and a ribosome. However, in eukaryotes, right, you've got the nuclear membrane. And so RNA has to be exported out into the cytoplasm before translation can begin. What else do you notice is different here? Yeah, we have this thing called pre-mRNA that's the product of transcription, and then something happens, right? We process it to produce the final mRNA that is used for translation. Right, so we're going to have to do some extra steps in eukaryotes that doesn't happen in prokaryotes. All right, so pretty straightforward when it comes to what's happening with our nucleotides. Right, so here's a piece of DNA, here's our gene, right? Now, when we're doing transcription, which strand is the template? Or are they both templates? How many templates do we have? Just one, right? How many templates did we have in DNA replication? Two, because you were making copies of both. So in transcription, we have to know which one is the template strand when we're looking at things, right? Your cell knows because of the regulatory sequences, right? So this is kind of a sneak preview for on Thursday, but I think it's helpful to put this in context. We'll call this the anatomy of a gene. All right, so here's my gene. Right, upstream just means on the 5' side of things. Right, so we're assuming we're going this way. Right, so over here is my promoter. This is what we call +1. This is where transcription of my gene is actually going to happen. Right? And then at the end of this, there's going to be some sequence that tells RNA polymerase when to stop. What do we call this? It's the terminator. Right, so the sequence of DNA here tells RNA polymerase, hey, there's a gene here, and this is the spot where you start copying. this DNA sequence is basically telling RNA polymerase, you're done copying, you're gonna stop. Right? So, right, RNA polymerase is gonna recognize that signal. It's gonna bind here, so I'm gonna write RNP for short. Right? And then it'll start copying here at +1. So we're only copying from here to here. This sequence and this sequence are important because they tell RNA polymerase what to do essentially. But they themselves are not transcribed. Okay, so we have 2 strands there. So not only does RNA polymerase does the promoter tell RNA polymerase start copying here, it tells it which strand is the template, right? It has to know which one do I copy. So RNA polymerase dictates which strand is the one that you're going to copy. Okay, so if that upper strand is our template, right? This is our RNA copy, right? So A is complementary to U, C, G, C, G, so on and so forth. So far so good? Nothing new there. The only thing is that we replaced the T's with U's because we're building RNA. Right? And then you'll remember when we translate, we read in groups of 3. We call those codons. Right? And so each group of 3 codes for a specific protein. Remember the SPIFFy decoder chart? Genetic code chart that you have, right? So you can look up the mRNA codon, and then you would find out which amino acid that is, right? So UGG would give us tryptophan. UUU gives us phenylalanine. GCU or GCG gives us glycine, so on and so forth. Right? So you're going to use the SPIFFy decoder table. We're going to talk today about how the ribosome does that. Okay, so nothing new here. You guys know the distinction between DNA and RNA? All right, so here's transcription. So here's a much better diagram from what I drew here. All right, so what is this sequence that you can see here where RNA polymerase is bound? That's the promoter, right? The promoter is where RNA polymerase sticks to the DNA. Right, so where it says start point, what is that on the diagram I drew here? That's that +1. Right, so that's where we're going to start the process of transcription. Now, that promoter is telling RNA polymerase there is a gene here that you're going to copy. So which strand, right, if this is the gene and we're going to copy going this direction, which of these 2 is the template?
The top one.
The upper one or the lower one?
The upper one. The upper one.
Raise your hand if you think it's the upper. Raise your hand if you think it's the lower. Raise your hand if you're not sure. Okay, so let's think about this. Let's draw it out. Right, here's my 2 DNA strands. So this top one is 5 to 3. This one's 5 and 3, and we're going to go this direction. So if I'm going to go that direction, right, I have to be building which way? That way. Which means this has to be which end? Which means this has to be which means it's complementary to this one or this one? The lower one. Right? Because they have to be in opposite orientation. Right, so the promoter tells RNA polymerase, hey, this is the one. If it told it was the upper strand, which way would RNA polymerase be going? It would have gone this way, so the gene would be on the other side. Okay, so there's 3 steps to this process. The first thing is initiation. We have to unwind the DNA, just like we did in DNA replication, right? So we got to pull the DNA apart, and then RNA polymerase can begin the process at the +1. Okay, now, what can RNA polymerase do that DNA polymerase cannot? What's it doing here that DNA polymerase couldn't possibly have done? It can start from scratch, right? right? We don't need a primer here, right? RNA polymerase is starting to build as soon as we pull the DNA apart to expose the template. Okay, so initiation, RNA polymerase binds to the promoter, unwinds the DNA, right, it creates the bubble, and now we're going to start. So elongation is now we're just zipping along, making a copy as we go. So as soon as it's done in that area, right, the DNA just winds back up, right? So that bubble travels as we go until we get to the termination sequence at the end of the gene.
You don't need
RNA polymerase has helicase activity, so it has the ability to unwind itself. Right, and then the third step, termination. We get to the end, and RNA polymerase is going to receive a signal. It's going to essentially fall off the DNA, and so it's going to stop transcribing. Right, so initiation, elongation, termination. Unwind the DNA. Right, create the bubble. We call this a transcription bubble as opposed to a replication bubble. Make your copy, fall off. Okay, so how does this process start in eukaryotes? Well, the promoter in eukaryotes has a sequence called a Tata box. Right, so if we looked at a lot of genes, we'd find a pattern that fits this sort of sequence, right? They're not all identical, but there's oftentimes, almost always, something like this Tata box. So it's a sequence that RNA polymerase recognizes, right? And that's why it knows that it's supposed to sit here, right? So the promoter region has a sequence in common that allows RNA polymerase to bind to the DNA at that spot, right? So here's our promoter region, right? It's upstream of our coding region. Right? Now, there are going to be other proteins that are going to help RNA polymerase stick to the DNA. And we'll talk more about those on Thursday. Right? But these things are called transcription factors. Yeah?
So the Tata box is in the promoter?
Yes. It's, so the promoter, might be very long. Usually it's a few thousand base pairs upstream. But then the Tata box is literally 10 bases long. It's usually very close to the start point.
It's in the beginning of the promoter?
I wouldn't say beginning, I would say it's very close to the start point of transcription. So RNA polymerase is going to recognize the Tata box, it'll grab onto the DNA here. Right? But there are other proteins, there's a lot of other proteins that are going to help RNA polymerase in this process. So we'll talk more about that later. Right, so once these transcription factors help RNA polymerase bind, right? So it's not just RNA polymerase recognizing the Tata box, right? It's these other factors that recognize the promoter and RNA polymerase together. So RNA polymerase isn't really capable of doing this by itself. It's got a bunch of helpers. Once they're on the promoter, though, then RNA polymerase is ready to begin to unwind the DNA. Right, so this is a process we call recruitment. Right, we recruit RNA polymerase to the promoter. Questions about initiation? Transcription factors help RNA polymerase recognize and bind to the promoter. Once RNA polymerase is bound at the promoter, we can begin.
So the transcription factors just kind of hover around the promoter region?
Your transcription factors are
Kind of
everywhere,
everywhere?
right? They just bounce around in the nucleus. But, right, if not all transcription factors are being made all the time, right? So if these transcription factors haven't even been made, we might not ever turn this gene on, right? So it's not until we have these that we would turn the gene on. Right, so it's one of the ways that we can regulate whether a gene is on or off, and we'll talk a little bit more about that on Thursday. Okay. Right, so if this is our DNA, right, the lower strand here is our template. So here's the bubble, right? And this is our new RNA. So RNA polymerase is just putting in each nucleotide, right? So here comes the A, it's complementary to the T. This is the 3' end, so we're going to build what kind of bond here? Phosphodiester, right? We just keep going. So this bubble, we just keep going this direction, and the DNA zips back up behind it, right? So the RNA message that we're making is just trying out the exit port from RNA polymerase. Okay, now, we should point out, there's nothing that says that only one RNA polymerase can hop on and then transcribe, and then another one can go on, right? You can have a gene being transcribed by multiple polymerases at the same time, right? One can hop on and it starts going, and another one hops right on right after that and starts going, right? So we can make a lot of copies from the DNA very quickly, right? RNA polymerase just starts and gets going, another RNA polymerase can hop on, and we can make a lot of copies, right? So when we talk about gene expression, you can kind of think of it like the volume on your stereo, right? I can have it on, but have it really weak. So I'm just making very few copies of a gene. So I don't make a lot of RNA. I could turn it up higher, right? So now I'm making more RNA copies. I could turn it full blast and make tons and tons of copies, right? So in one way that we can regulate things is how much transcription are we actually doing? Because, right, in theory, once we've made the RNA, we have the information to make a protein. This step, the initiation step, not elongation, the initiation step. This is the single most important step in regulating gene expression, right? Because as soon as I turn on the gene, I've made the RNA, right? After that, I can make protein, right? There's lots of steps that we can regulate, but if we never turn on transcription, I'll never make a protein. So this step, initiation, is the most critical in regulating whether a gene is being expressed or not. Okay, so elongation, RNA polymerase is zipping along, making its copy until we hit the termination sequence. Right? There are different ways that genes can be terminated. Right? One is self-termination, where the termination sequence has a sequence where the RNA makes this thing called a hairpin. So hairpin would look something like this. Okay, now why would RNA make a structure that looks like this? Well, if I told you this was an A, and this was an A, and this was an A, what would that be? It'd be U's, right? Because this is RNA. Right? So if I have a sequence where it can fold and pair up like this, we can get this thing called a hairpin structure. Right? And then, you know, maybe you have something like this, right? So there's not really the ability to base pair very well here, but here I have enough stuff that I can make this structure. Right, so when this happens, it kind of folds up inside of RNA polymerase's active site and forces it off. Right, so it's kind of bumping it off as it makes that RNA, the RNA folds up, and it bumps RNA polymerase off the DNA. Right, so that would be, right, the sequence in here. In prokaryotes, right, prokaryotes do something else. They have an enzyme called Rho. And Rho does this thing where it starts climbing up the RNA message as RNA polymerase is going along. And when Rho catches up to RNA polymerase, it causes RNA polymerase to fall off. It sort of pushes in between RNA polymerase and the DNA, causing it to fall off. Right, so this protein attaches at the beginning of a transcript and moves up. So it moves up at such a rate that it forces this off about the same point where it's supposed to stop transcribing the gene. So two different ways. These aren't the only ways, but these are some of the more common ways that genes can be terminated.
on the termination side?
Correct. So it's been zipping along, right? And rho reaches here at the termination sequence. So this is what we call rho dependent termination, right? There's also rho independent termination where you don't have to have rho, right? This would be an example of rho independent termination. Right, so in either case, you're physically forcing DNA polymerase to fall off the DNA. One physical way is by making a hairpin. The other is by a protein that wedges itself between the polymerase and the DNA. Okay, a few things to note. First of all, remember we said that we have different types of RNA molecules, mRNA, tRNA, rRNA. Another one that we're going to learn about here called snRNA. Right, these are all products of transcription. Right, I took some DNA, I used RNA polymerase, I made a copy. But they are not translated. Right, the RNA molecule itself is sufficient in tRNA, rRNA, and snRNA. Right, mRNA is translated. So a little misleading the way I worded it, right? mRNA is translated, but tRNA, rRNA, and snRNA are not translated. Okay, so we'll talk more about transcription factors on Thursday, so don't worry about those. Remember, only one of the two strands is the template, right? So that's the strand that will always be the template. Remember, the promoter is the sequence of DNA that's going to set where the gene is going to be, right? So where do we begin transcription, which strand it is, and then how much. This is the other thing we didn't mention so far, right? The promoter is like the volume control, right? I can have a promoter that we would call a weak promoter. It turns on transcription just a little bit. Right? Or I could have a really strong promoter, something that turns on transcription like full blast. Right? So a lot of times if we're doing stuff in molecular biology, I might take a promoter from a virus because a virus usually has like a really strong promoter. It's really good at getting transcription to do its thing. Right? So maybe I'll take that and I'll put that in front of a gene that I want to express in a cell so that I can make lots and lots of transcript. Right? So the promoter dictates where the start is, right? Which strand, and how strong transcription is going to be. We're going to get back to this, but you want to make a note to yourself, highlight it in bold, whatever you need to do. You need to make sure that you can distinguish between the transcriptional start site, which is +1. So you'll see this term used as well, TSS, or transcriptional start site. You do not want to confuse the transcriptional start site with a start codon. And we'll try to illustrate this later here in a few. But make sure when you go back to study, you can distinguish between those two. It will be critical.
So the beginning of the codon is going to be the beginning of the promoter, right?
No. The codon has absolutely nothing to do with the promoter or transcription. Right? Codons are solely for which process? Translation. Right? So the codons have nothing to do with RNA polymerase or transcription. So we'll see where the codons are relative to this in a few minutes. Remember how DNA polymerase can fix its mistakes? At least some of them, right? It has proofreading. RNA polymerase basically doesn't fix any mistakes it makes, right? Very poor proofreading, right? Do we care if RNA polymerase makes a correction for every mistake? Probably not, right? And the reason why is because if I make one mRNA copy and it's not that great, we're just going to make more copies, right? It's not the permanent version of the gene, right? So it's like if somebody was making a photocopy and there's a smudge. Am I concerned that the photocopy has a smudge? No. I just go back to the original, make another photocopy. right? So we don't try and fix RNA, right? We don't try to edit RNA the same way we try to fix our DNA, so correct DNA. Okay, so if we're talking about a prokaryote, as soon as we've made the RNA, we can translate it. Now we got to talk about this stuff. What's going on in eukaryotes? Because eukaryotes are a little bit different. When we make the mRNA for a eukaryote gene, there are different regions, and only some of those regions actually code for protein. Right, so we're going to break this up into coding and non-coding regions called exons and introns. We have to get rid of the stuff that doesn't code for protein. Right, so this is our pre-mRNA message. We're going to have to process this thing. Now, the end here and the end here are not necessarily start and end of the information for making our protein, right? We have a region where we're not going to translate anything. And we call this one the 5' untranslated region, or UTR, because we're not going to translate any of this. The start codon is not right at the end. Right? The start codon is somewhere in here, but it's never just at the end of the RNA. At the same sort of way, we have a stop codon here for translation, but it's not at the end of the molecule. So we have another region at the 3' end of our molecule that we call the 3' untranslated region, right? That doesn't get translated. So somewhere in between here is the information that we're going to translate. So we're going to have to do a few things to process this RNA before we can translate it. Right? The first thing we're going to have to do is add a 5' cap. Right? This is a specialized guanine nucleotide. Right? It's basically like a upside down and backwards guanine. It's getting chemically added to the end. Right, so all you have to remember is that it's a 5' cap. It's a modified guanine. The reason we add the cap is because it protects from exonucleases. What's an exonuclease? Something that cuts up DNA, right? It hydrolyzes DNA, but it does it from
Outside the nucleus.
from the ends, right? Something that chews up from the ends. An endonuclease would be something that could just cut in the middle, right? So an exonuclease, right? You've got lots of exonucleases running around in your cell, and that's normal because we need to get rid of some stuff, right? We're going to recycle mRNA all the time. So when we make an mRNA, it's only going to stick around for a few minutes or a few hours for the most part, right? And then it's going to get chewed up. And if we need more, we just do transcription again, right? So an exonuclease would just chop it up. But, right, that 5' cap will prevent this thing from being chewed up right away. So as soon as we make it, we stick a cap on there so it doesn't get digested. All right, the next thing that we're going to do is we're going to add a poly-A tail. Right? So on this end, we're going to add usually 200 to 300 adenine nucleotides. I'm just going to make a whole string of them on the end. Right? Similarly, this helps to protect from degradation. So degradation just means it's being broken up or broken down, hydrolyzed. Right, so both the cap and the tail are going to prevent our RNA from being degraded before we can translate from it.
So is the poly itself bound in the 3' translation?
Yes.
So
It gets tacked onto the end.
Okay.
And so it's always going to be in the UTR because everything after the stop codon is not going to be translated.
And then the 5' cap is just attached to the 5'
It's right at the 5' end. Right, so here's, well, there's your poly-A tail.
Oh,
Right, so this is the signal. So the 3' UTR, I mean, all of this is untranslated, I guess is the way to think of it. Right? The 3' UTR has some signal that tells the the enzyme to add the polyazole. Okay, so we're gonna have 5' cap, we're gonna have polyetail. And the last thing we're gonna do is we are going to splice out these things called introns. So we have introns and exons. An intron is an intravening sequence. It's a sequence of DNA that does not have the information we need to make our protein. So we're gonna get rid of it. Okay, so here's how this works. Right, so here's my pre-RNA, pre-mRNA, here's my 5' cap, my poly A tail, right? And then I have, in different colors, introns and exons. Right, so for example, this region, and this region are my exons. So this, and this have the codon information to make a protein. But this region here and this region here do not. So I want to cut these out. I want to cut out the introns, and I want to splice together my 3 exons, right? So 1 through 30, and then attach 30 to 31, and then attach 104 to 105, right? And get rid of the stuff in between. So that when we're done, we have this. This is my final mature messenger RNA. All I have are my exons. I have no more introns. Prokaryotes don't do this. Prokaryotes do not have introns and exons, right? Everything in the RNA codes for protein. Right, so we have to do this thing called splicing. Okay, so how does this happen? Well, we have a thing called a spliceosome. So a spliceosome is a bunch of proteins, right, along with these snRNAs, right? So those snRNAs help the proteins recognize the boundary between the exons and the introns. And so what it's going to do is basically make a loop to cut out the intron and then attach at those 2 boundaries together the exons. Right, so the spliceosome is going to splice out the introns, splice together the exons.
How does it know what's an intron and what's not?
So at the boundary, there's usually a common sequence. So if I took a bunch of genes and I looked, right, I could pick out a pattern that shows up at that region. And so, right, my RNA is complementary to that, so it helps the spliceosome recognize, hey, this is where I need to hydrolyze. And the same thing over here, it'll recognize what it needs to
So
hydrolyze.
all introns have that characteristic?
Yes. So at the intron-exon boundary, you'll find this pattern. And in fact, if you have a mutation here, right, that'll be a problem because then you wouldn't cut out.
Where do the entrons go? Like, do they just get recycled?
Yeah, so now this is just going to be degraded, right? So we can chew that up, and now I have my building blocks to go reuse and make RNA again.
The S on the snRNA is called spliceosome.
You want to know something really sad? It's been so long, I actually don't remember what SN stands for.
Small nuclear.
What's that?
Small nuclear.
Small nuclear, there we go. Things I should know. I should not be forgiven for that. It's been a long time since I've looked at that acronym. So, you know it's snRNA, I'm happy. A lot of acronyms. Right, so you should be able to distinguish between snRNA, tRNA, mRNA, rRNA.
Just to clarify, snRNA helps in aiding of cutting out the introns, right?
Right, it's a part of the spliceosome.
By the
So just like a ribosome is made up of rRNA and proteins, the spliceosome is made up of snRNA and proteins. So they function together. Okay, now, I don't know about you, but I'm sort of asking myself, if I were in your shoes, why the heck are we doing all this? Right? Like, doesn't the prokaryote have the better idea? As soon as I'm done with transcription, I'll just go translate it. Why do all this extra stuff to cut out this extra material? Why? The simple answer is, remember you have about as many genes as a chicken? Or fewer genes than an amoeba? It doesn't matter how many genes you have, it's what you can do with them. And just having 20,000 genes doesn't mean I can only make 20,000 proteins. I can make a variety of different RNA, messenger RNAs from one gene. And if I can make different mRNAs from one gene, what else can I do? I can make different proteins, right? So this is called alternative splicing, right? And most genes in humans, for example, have several different, what we call alternative splicing isoforms, right? So here's our DNA, right? And in our gene, right, what would this be right here?
The TS.
Transcriptional start site, +1, right? So that would be where we start, here's where we end. Okay, so this is just an example of a specific one called troponin, right? So troponin has 5 exons. So how many introns does it have?
4.
1, 2, 3, 4? All right. So if we transcribe this, right, I get this whole thing is our pre-mRNA. Now we're not showing you what's over here. What should be here that we're not showing you?
The stop.
Poly A tail. What should be here? Gap. Okay. Now, there's nothing that says that I have to go 1, 2, 3, 4, 5. Right? Depending on the needs of the cell and what it's trying to make, it might splice together 1, 2, 3, and 5 and skip out 4. Right? Or it might do 1, 2, leave out 3 and do 4 and 5. Right? So it's got alternatives when it splices. Hence the name alternative splicing. Now, the sequence in these exons is the same, right? So, right, the proteins that we would make from these two would be very similar, right? Because these two sequences are the same, those are the same, these are the same, right? What would be different would be just whatever information is coming from the codons that are different. So these would be very similar proteins, but they wouldn't be very identical, right? If we translated these, the proteins might have similar functions, but they wouldn't be identical in function. So I can have one gene, but make multiple messenger RNAs, and from that I can have multiple proteins, each with a slightly different function.
Could you ever just get pure ones in the RNA splicing?
Say that one more time.
Could you ever just get like 4 of the ones, or?
No, because you only have one here, right? There's only one copy of one.
Oh, okay.
And you would so that's a good question. And the other thing is you would never scramble them, right? I couldn't do like 5, 3, 4, 2, right? It does it in order. So I will always go in order, but I don't have to have every single exon there.
Can I do like only 1, 2?
Sure. Although oftentimes the last exon is included because that has the stop codon inside of it. Yeah. Not always. Okay, so, right, if we've got 20,000 genes in humans, we can make, 100,000 different proteins. Well, right, so, on average, the average human gene, I think, has 4 or 5 different splice variants. So we've got a lot of possibilities for proteins, even if we only have 20,000 genes.
Do prokaryotes do the alternative splicing?
Prokaryotes don't do any of this. They don't do splicing at all. They don't add a cap. They don't add a tail. So they don't do any RNA processing whatsoever. So we spend a lot of energy and resources on this process, but it pays off, right? This contributes heavily to the complexity that we see in eukaryotes versus prokaryotes. Right? Now, the other thing that I want to point out here that we didn't mention before about proteins is proteins are modular. Right? So exon 1 gives me a specific sequence. Here's where I get from exon 1, right? So we would call this a domain. So exon 1 is always going to fold up in this particular shape, right? And then exon 2 is going to give me this. Exon 3 will give me this. So usually, right, the three-dimensional thing we call a domain, they each have different functions. Right, but then they're all sort of stitched together. So even within an individual protein, different regions might have different functions. So we call those regions domains. So if I had these two different splice variants, right, the domain 3 is different than domain 4. So domains 1, 2, and 5 are the same. So a lot of what this protein can do would be conserved between the two proteins. But because domain 3 and 4 are different, right, this protein is going to do something the other one couldn't. Right, because this is going to fold up, it's going to have that particular shape. Right. So one thing we could do, right, and this is one of the ways we think that we get new genes, is we can, sometimes there's mistakes in DNA replication, and we accidentally make multiple copies of certain regions, right? So then I can have extra copies of exons. Sometimes things get shuffled around, either because of viruses or other errors during replication. We might show this later. And so we kind of, it's like taking these things and just shuffling them around, and we get a new combination of exons. We might get some exons from this gene and some exons from that gene. And now what we have is a new combination of exons, i.e. a new protein. And because it has these different combinations of exons, or different domains, it's going to have a different function from either gene that they came from originally. So this is one way that new genes are generated. Okay, so we've talked about this before, right? The distinction between mRNA, ribosomal RNA, and tRNA. Okay, so a few key differences between prokaryotes and eukaryotes. Remember we said that transcription and translation can happen at the same time for a prokaryote. As soon as we make the RNA, the ribosome can hop on because there's no nuclear envelope, right? That doesn't happen in eukaryotes. We have to export the RNA out of the nucleus to the cytoplasm, right? There are no introns in prokaryotes, right? But eukaryotes have introns. There's no RNA processing in prokaryotes. We have to do the poly A tail, 5' cap, and splicing in eukaryotes. And the last thing is the RNA that we produce from eukaryotes is what we call monocistronic. All that means is that one RNA molecule codes for one protein. On the other hand, in prokaryotes, the RNA could be polycistronic, which means that we can encode multiple proteins on a single RNA message molecule. And what would this look like? And we're going to look at a polycistronic example here on Thursday, so just put this in perspective. In a prokaryote, what I'm going to have is a single promoter and then I can have multiple genes after that. So, right, here is LAC-Z, LAC-I, LAC-Y. I don't even remember if that's the right order. Find out Thursday when we look at these things. So I got 3 different genes, right? But I only have one promoter. So when I get the RNA for this, I get an RNA for all 3, right? LAC-Z, LAC-I, and LAC-Y. So then a ribosome is going to hop on here and make one protein from here. And a ribosome is going to hop on and make another one from here. And then another one from here. Right, so one RNA gives me 3 proteins. That's not the case with a eukaryote, right? Prokaryotes are polycistronic, right? If we were to transcribe this, I would just get, this is gene X. This is just gene X's mRNA. That's it. So I'm only going to get 1 protein. This whole thing, where I have 1 promoter and multiple genes together, is called an operon. And we're going to look at operons on Thursday, because it's a really good example of how we control gene expression. Okay. Hey, come on in. Let's take a breather.
Hello, everyone. My name is Victor. This is Holly. We're tutors in LRC. So we just wanted to give you guys a brief announcement about our workshops. We do workshops trying to every Friday, but specifically we're doing a workshop for your class this Friday. We haven't yet come up with a time and a specific location, but the location is the LRC. We just don't have a specific room. As far as the topics, we believe you guys have a test next Tuesday, right? All right, so as far as topics, you guys are going to be coordinating with your professor and with Holly here, because she's going to be running that workshop. So what you guys are going to do is, if you can, I believe the professor is going to, how are we going to go about this?
I'll shoot out an announcement. I'll probably post either a discussion or have you guys just email me directly or Holly directly. Maybe I'll just do a discussion, because then you guys can just toss in whatever you're looking for. We have a record of
those,
what everybody wants.
Perfect. So those topics are basically going to be about what you guys think is going to be on exam, or what you guys are probably having trouble with in your class. And we suggest that you guys try to get that to Holly, even if you can in person, as soon as possible, because it gives her time to study up on this bit and try to give you guys a good explanation on what you guys are probably confused about. So the workshop is really good to go to this Friday. We suggest you go just because it helps you really, if you guys are shaky on a few things, and it helps you to really solidify what you know and what you don't know. That's why I like to go to workshops, because talking over it with someone else helps me to really understand, okay, I'm really good at this topic, so I'll skip that when I'm studying, and I'm really shaky at this, so I'll focus on that. So we suggest you go, and we'll try to get more information to you via your professor before this Friday about the specific location. Right now, it's just in the LRC. Do we have time yet?
Between 10 and 4?
Anytime between 10 and 4, because the LRC is open if you just want a general tutoring between 10 and 4. The LRC, if you guys don't know, is a library. If you don't know where the library's at, it's a big water fountain on the main campus. It's right there. All right, so yeah, just come see us. We'll try to get back to you, hopefully, I hope Thursday with a specific time and room number, but for right now, it's just the LRC. Come up with any topics. Topics can be like cellular respiration, or if you have a specific question, put that specific question, but try to make sure you get that somehow in Holly's hands before, so she can come up with your specific answer. And yeah.
We'll try to cover as much as possible, but we're probably focusing on the ones that multiple people say. So if you agree with what someone else posted, like, can they like it? Yeah, like it. I don't know how to work.
Yeah.
Or just be
Parentheses
like,
like.
yes,
Yeah.
this one.
Yeah. Something like that. Yeah. So yeah. Anyways, yeah. Just let us know if you have any quick questions. Nope. All right. Yeah. LRC Friday.
Thank you.
Thanks, guys. When they get the info, I'll shoot you an announcement so you know the time and the place as well. Thanks, guys. All right. So, translation. You guys are aware of how to use the genetic code test, genetic code table, right? So you'll notice it's redundant, right? So I have multiple codons for histidine, but CAU like only gives me histidine, doesn't give me anything else, so it's not ambiguous, right? So run through here, right? Again, I would highly recommend that you get some practice with this. Right, if that's our thing, then we looked up each codon. Right now we know what our protein looks like. Right, same thing if I gave you this, right, which end are we gonna start from here, from our, alright, so you're always gonna start from the 5' end, right, whenever you're translating, you go from the 5' end, right, so you're gonna find your start codon, right, now it's not necessarily always the first start codon, but for us it will be, more things that play into that, but for all intents and purposes for us, as soon as you find the start codon, that's where you're gonna begin. So then you mark off your groups of 3, right? So I just want to go until I find what? Stop, right? And then I'm going to look up each codon on my RNA chart. All right. So how is your cell doesn't have a spiffy coder? You know, your ribosome's not looking at this code table and being like, okay. So how does it do it? Right? Did you guys talk about how the ribosome interacts in lab on last week? We did in the morning. Did you guys do it in the afternoon? Did you guys talk about the ribosome? Okay. Remember that thing I told you we were going to see over and over and over and over and over again? The hydrogen bond? Right? The only reason this works is because of hydrogen bonding. Right? So here's your ribosome. Right? Your tRNAs are carrying amino acids. Right? And your tRNA is an RNA molecule. So guess what it has? It has nucleotides. So those nucleotides are complementary to the codons on your mRNA molecule. Right? So your messenger RNA is going to be complementary to each different tRNA. Now each tRNA is going to carry a specific amino acid. Right? So it's a one-to-one relationship. One tRNA has one anticodon, which is what we call this region, that's complementary to a codon, and they're each carrying a single amino acid. All right, so this is our tRNA. This is the generic shape we're using, but it's an RNA molecule, right? So it's going to fold up into some three-dimensional shape because it can base pair with itself. Right? So all your tRNAs have the same basic shape. Right? We're going to add the amino acid to the 3' end. And then the anticodon is on the opposite side. Right? Now, you have to have some protein that puts the amino acid onto the tRNA. Right? So the aminoacyl tRNA synthetases are going to recognize, hey, this is a particular tRNA, so I need to add tyrosine to this one. How does it know that? Well, you have the anticodon, right? So that anticodon is specific for the amino acid tyrosine, and it fits in here, right? It's got the right shape, so it's a combination of those two things. So a tRNA is going to pick up an amino acid. It's going to get loaded with that amino acid by the aminoacyl tRNA synthetase. And then where is it going to take that amino acid? Once I've attached it, what am I going to do with that? Where is this thing going? Now that I have a tRNA with amino acid.
To the mRNA strand.
Yeah, you're going to take it to the ribosome for translation. Right, so now that we've made this, now we can go to the ribosome. Okay, so here's the quick translation, right? The first thing that's going to happen is the start codon is the first tRNA that's going to be recognized. So your ribosome has a small subunit and a large subunit. They have to come together. So first your small subunit attaches with your initiator tRNA, which has the anticodon that matches your codon. If it's the first one, it's always carrying the thymine. Okay, so once these guys attach, right, this part of this ribosome can attach, and now you're ready for this process to begin. Right, so you have 3 sites, the E, the P, and the A site. Right, so the initiator tRNA is in the P site. So if I wanted to attach another amino acid here, how would I get it there?
You would have to add
bring a tRNA here?
Oh, you create a bond.
I wanna make a bond, but how do I get a tRNA here first?